Hi folks, Jim here. What I've got for you today is part 1 of the origins of the COVID-19 causing virus. Specifically, we're going to be looking at the viral genome and how you trace a pandemic back to its original starting point. This was recorded live on Twitch on the 29th of June 2020 and so was accurate at the time of recording. I'll link parts 2 and part 3 in the video description along with my Twitch channel. You can follow me over there to see live broadcasts of science related content on Monday, Wednesday and Thursday evenings UK time. I hope you enjoyed the video. If you have any ideas or suggestions, leave them in the comment section or hop on over to Twitch and you can talk to me live during one of the broadcasts. Let's roll the video. All right, I think we're on. I'm just going to do a quick monitor test. And we're good. Right. Yeah, I had a few problems with my mic in the last stream. So a bit of a different format from last time. This is going to be me just on my own, scary stuff. But I wanted to go through today was actually prompted by a conversation that I had with a mate of mine called Andy. And if you're watching, thank you for sending me down the rabbit hole of this, what today's little talk's going to be about. So it says it on the screen. We're going to be discussing the origins of the SARS coronavirus 2 virus, effectively. And what I thought I'd do today was, or this evening, in fact, was I just drew up a little Google Docs document, put some headings on that we'd like to talk about, and then we can add notes to it just as something to follow. Really informal. I don't know how long it's going to take to go through this. Some of the scientific literature I've already seen. Some of it's fresh. But effectively what I want to do is go over, give a quick overview of the infection, more just for background really, discuss a couple of terms that might pop up in the literature, which sometimes can be a bit confusing. And then we'll get into the meat of it. So really, talk about how do you actually track an infection? And if you want to find out where, say, a virus comes from, how do you do that? We'll follow the outbreak timeline, the locations, which I think are fairly evident, these two. Animal infections and animal reservoirs, that's really important. And that will become really obvious as we go through it. And genetic tracing, which will actually form kind of the crux of of how we actually do the tracing. And at the end, what I've done is what don't we know? So let's go through what we don't actually know and what's still kind of a mystery and still to be solved. And then get on to the two tin hat topics, which are, was it released from a lab? Is it a bioweapon? And I'm probably not even going to get to go into 5G because bloody hell. Right. Some people are complete idiots. Right, anyway. So if you want to comment and ask questions, you can do. I've got the chat here. I will aim to look over at it as much as I possibly can, but generally I may forget. It's no offense. It's nothing personal. So let's go. Right, so an overview of the virus infection. So in order to kind of track where the coronavirus actually comes from, you have to kind of understand about its transmission. So something to note about the coronavirus, and one of the reasons why it's become such a global pandemic, is this idea that you have a relatively long incubation time. Now, so an incubation time, get a nice color. So the incubation time is effectively the time from when you are infected to the time where you display symptoms. And so the reason why that's important is because you probably don't know you're infected until you display symptoms. But you could be contagious. So you could still pass on that infection. So the incubation time for COVID-19 is, I believe, it's around, it's about a week. Where you could have picked it up, you could be infectious, but you wouldn't know about it until a week later when you start displaying symptoms. And the reason why that makes it quite a tricky virus to trace is because generally your first port of call when you're tracing a virus is you're tracing the people that are infected. So what might happen is that you've got to go and trace a week's worth of interactions, right? So that's a lot of people. Can you imagine if you were forced to write down every single person you've interacted with in a week? You'd probably miss some people. So this is very important in terms of tracing back people. And it's very difficult to do. So fortunately, that has kind of been done already. So we knew you can do it by like geolocation. So we knew, scientific community knew that there was an outbreak in China around December of this new pneumonia. There's actually patients going into hospital with pneumonia. Got a really good paper of it, which is this. So this is effectively an article in the New England Journal of Medicine, very highly reputable scientific resource. In fact, why don't we first quickly, before we get into that, let's quickly do the terminology, because there's bound to be some things in here that are a little bit sciencey and a bit nuanced. So we'll do first things first, right? So COVID-19 is the disease. It's not the virus name. This is a disease that people get. The virus is called SARS, stands for Severe Acute Respiratory Syndrome. Coronavirus, that's the abbreviation for coronavirus 2, because there was a SARS coronavirus 1, and that was discovered in the 2003, I think, outbreak of the original SARS virus. So this is SARS-2, or SARS-CoV-2. You might also hear that called, oh, bad typing, N for novel, sorry, N, COVID-19. You might see it written like that. That stands for novel coronavirus, and that just means it's a new coronavirus that was discovered. Effectively, commonly, these are all interchangeable, but let's just get the terminology down before we go into this. So yeah, if we go back to this New England Journal of Medicine, this is a free paper. You can read this for free. All you have to do is type in the title, or you can just go to the New England Journal of Medicine. And you can, effectively, if you just read the intro, right? Let's have a look. Might not be the intro. What I don't want to do is I don't want to spend too long on this, but I will kind of skim it. Right. So in late December 2019, several local health facilities reported clusters of patients with pneumonia of an unknown cause that were epidemiologically linked to a seafood wet animal wholesale market in Wuhan, in China. Right, that effectively means that these several people that had this unknown pneumonia, they were asked, where have you been in the last 2 weeks? And I think out of the original cases that were reported, I believe it was 60% had all had this wet seafood, this wet animal market in common. So that's quite a high percentage of people, and that is effectively drills down on where they may have all contracted this from. So on December 31st, the Chinese Center for Disease Control and Prevention dispatched a rapid response team to conduct an epidemiological and etiological investigation. And this paper reports the results of this investigation, identifying the source of pneumonia clusters and describe a novel coronavirus detected in patients with pneumonia whose specimens were tested by the China CDC at the early stage of the outbreak. So this paper effectively goes over how they detected the virus. So if we go back down to here, we're talking about tracking an infection, right? So the first thing you do when you track an infection like this is you look at geolocation. So, where did this first erupt? Now, fortunately, in today's day and age, what we've got is a very interconnected world. So you can imagine that if back 100 years ago, during like the Spanish flu, what would have happened is that there would have been pockets of this flu erupting, but then no one would have really known what it was about. If you were living in London, for example, and someone has flu in Madrid, you're not going to find out. But today, because of the interconnected world, the internet, social media, you do find out the locations of infections relatively quickly. Probably not as quick as we would have liked, but the geolocation is your first port of call. So we knew that this was from Wuhan, and it was in December 2019. This is the reason why it's called COVID-19, because it started in 2019, despite the fact that pretty much everything around the world has happened in 2020. It was actually discovered in 2019, just at the back end. So that's why it gets the designation of COVID-19. So you're tracking people, right? This is the first thing that you track. So you can track everyone to this wet market. And that's fairly established. What we found out since the wet market, and I think that's in this, what we found out since then, it might not be in this paper, but I think it's probably in a different one. But what they effectively showed in this one was they're isolating the virus from the known patients. So what you do is generally, if you have an unknown infectious agent that your regular tests can't find, you examine the DNA or the RNA, or effectively the genome, if you want to call it that, of this novel entity. And the way that they do that is, for this, they use something called bronchoalveolar lavage. Effectively, this is a sampling technique where they take the fluid that lines your lungs. That's called the bronchoalveolar lavage. Lavage fluid, I should say. And you can extract that, and that that should contain your infectious agent. And then what you do is you isolate the nucleic acid or the genome from this agent, and then you run what's called a PCR reaction. And the PCR reaction aims to amplify that nucleic acid, so you end up with quite a lot of nucleic acid, and then you can sequence it, or you can use other methods to detect a potential pathogen, right? So extracted nucleic acid samples were tested for viruses and bacteria by PCR using this kit and this piece of equipment. In accordance with the manufacturer's instructions, samples were analysed for 22 pathogens, 18 viruses, 4 bacteria, as detailed in the supplementary appendix. In addition, unbiased high-throughput sequencing described previously was used to discover microbial sequence not identified by the means tested above. So what you have is you actually have these test kits that will identify 22 known pathogens, so 18 viruses and 4 bacteria that are known to cause pneumonia. And then in addition to that, they sequenced the genome of whatever's in the alveolar lavage fluid, and they found in case there was something new. And indeed, this case, they did find something new. which was a little bit worrying. So that is the way that they've effectively identified a novel virus in patients at the back end of December 2019. So we know that they've got something new. We know that there's a cohort of patients that have all visited this market. So the next step is actually to go to the market and see whether or not there is any evidence of that virus that was in those people at that market. Now, I don't have a paper on this, but I have effectively, yes, they found the genomes and samples of the viruses at the wet market. So I'm using wording very carefully here because you generally don't find a live virus whole outside of a cell, and effectively it's to do with the fact the viral mechanisms. Viruses require cells to replicate. It's the reason that a lot of viruses don't survive on surfaces for too long, is because effectively they can be degraded by the elements, and they're not in the safety of a cell, they're not in the safety of a host. So effectively what they did is they went to the wet market and they tested the market and the surfaces around the market and the animals in the market for the presence or absence of the genetic material from the virus. Looking for the same sequence effectively that they found in the patients. And they found it. They found loads of it. So, wet market covered in virus. Interestingly enough. Okay, so now you've got a little bit more information to go on. Okay, so what do you do then? So you know you've got effectively you've tracked it down to the 10 patients, for example. You've tracked all of them to the market. All of them went to the market, so it's likely that they all picked up the virus at the market. So the next question is, well, how did the virus get to the market? Right, if you're tracking back. And this is where things get a little bit hazy. This is where we aren't fully sure of everything. So effectively there is a gap or a question. How did the virus get to the market? Right, so this is an important thing, right? So let's underline that. So you can look at this from you can actually come at this from the other direction, right? So you have the sequence of the virus. So instead of trying to say like location based, how did the virus get to the market, which is actually going to be very difficult. If you imagine all the different animals and trades and foot flow of all the people that are coming in and out of that market every day, that's going to be really, really difficult to actually kind of track where this has come from. It might almost be impossible to track where this has come from. So what you can do is, because you've got the sequence of that genome, and let me show you this, right? So this, you can see this. This is actually the genome of those isolates, right? So this is SARS, coronavirus 2, isolate, Wuhan 1, complete genome. This bloody long, well, I say it's long, it's not actually very long in terms of genetics. You can have a look at this and you can compare it to other genomes. You can compare this to other viral genomes that say aren't in humans, or you can compare it to genomes that are in humans, and you can see whether or not the virus has it mutated from like an animal virus. I mean, this is a wet market. This is full of live animals, so it's possible that some animals were at the market and they have a virus within them. The virus doesn't really affect them too much, but then it mutates and suddenly now it becomes super infectious to humans. Right? That could happen, and the way you would go about looking at that is you would you would compare this sequence against other sequences from other viruses. And that's what they've done. And that's what a lot of the research around the origins of this coronavirus is actually, and it's geared towards. So let's find. So there's a lot of information on this. Yeah, we'll go on to that bit later. Okay, right. So let me find. Where's that paper that I wanted to show you? I closed it. I have closed the window, which would be a shame because, oh well. Okay, so this says it in the text actually. So this is a paper published in the journal Cell. Again, like New England Journal, this is a very reputable source. So they've looked at it again like a genomic perspective. So what they've effectively done is they've gone and compared that genome to other genomes. And the thing about cell papers is they're usually absolutely humongous. This one's not too bad actually. It was okay. So what they have shown, and let's find this bit. Okay, so cases began. Okay, comparisons. Here you go. Comparisons between SARS-CoV-2 and other coronaviruses. Right. So the earliest genomic sequence was made. That's effectively what we've seen. We know it's a member of the beta coronavirus family. Okay, here we go. Right. Initial comparisons revealed that SARS-CoV-2 was approximately 79% similar to SARS-CoV-1 at the nucleotide level. So this is comparison between this virus that's causing this pandemic right now versus the one that caused the pandemic back in the 2000s. Okay, and there is actually quite a lot of difference. 79% in terms of a genome is a massive difference. You've probably heard the old adage that we share 50% of our DNA with a banana. Now, I'm not sure if that's actually true. It's probably not. But, for a species difference, you're not looking at anywhere, you're not looking at anywhere near 79% difference. That's a huge difference, to be honest. So what they're effectively saying here is that it's very unlikely that it's the original SARS-CoV-1 virus. So that's one thing to put down. That's a good. That's something that we need to kind of know. Has it mutated from the original SARS virus? Probably not.